Now, let's revisit graph representations of metabolic networks. We did discuss this a few classes ago. So, how do you convert a metabolic network to a graph? You can build a substrate graph where you have glucose. So, this is just a couple of reactions from glycolysis that you see here, where you have glucose plus ATP giving G6 phosphate plus ADP. So, it is maybe a little difficult to discern this because this graph is laid out for convenience, right. So, you see that glucose and ATP are connected to glucose 6 phosphate and ADP. The one danger here is that you might conclude that you can convert glucose to ADP and ADP to pyruvate. You may recall this example from the class a few weeks ago where we talked about reconstruction of metabolic networks, and I showed you how there is a direct edge from glucose to ADP and ADP to pyruvate. That is not shown here because I have restricted it to just the first few reactions of glycolysis, but essentially your nodes are metabolites and your edges connect metabolites participating in the same reaction, essentially reactants to products. But the thing is, you try to connect all reactants to all products because it sort of makes sense, but you can try to avoid doing that as well. Can you only connect the main reactants to products? That is a possible way to go about this as well. But sometimes it becomes difficult to discern what is actually the main reactant, but you might remember from our discussion on this retrosynthesis approach of reaction miner that there is a way to use subgraph edit distance and so on to identify what is the main backbone chain that is getting transformed in a particular reaction, right. So, in a reaction of glucose, I am going to call it G for simplicity, plus ATP giving G6P plus ADP, the main backbone transformations are this. I mean, and I would not call ATP, ADP a backbone transformation, but because glucose is the center of action here, ATP and ADP are like helper molecules in a sense. Okay. So, one option is a substrate graph. You can also have a reaction graph where you have R1 going to R2 going to R3, where edges connect reactions that share the same metabolites. You can also have a bipartite graph. This is a lot more descriptive, and of course you see here that there are multiple nodes for ATP that are shown for making this graph look less cluttered. But then you clearly understand a few things from this graph. I am repeating what we discussed a few weeks ago, that you need both ATP and glucose to produce both G6P and ADP. And now you cannot just say that convert glucose to ADP and pyruvate, because you see this edge that is really important for this reaction to actually happen. Okay. Recall, because this is a bipartite graph, there are no connections between the metabolites and metabolites or reactions and reactions. All reactions, all edges go from metabolites to reactions and out come new metabolites. You can simplify substrate graph by eliminating these currency metabolites, and you get this kind of thing, and you will, after several steps, you will get pyruvate as well, right. So that way this makes a lot more sense. So this is a pretty useful representation, the substrate graph, once you remove currency metabolites. But then sometimes you may not be able to identify what the currency metabolite is, and for example in an ATP synthesis reaction, ATP is no longer a currency metabolite. So you cannot just make a sweeping statement that any high degree metabolites or any metabolites that are participating in too many reactions, or handpick ATP, ADP, NAD and so on as currency metabolites. So it is prone to error. So how do we get around these issues? So we came up with a very nice algorithm called MetQuest to identify these microbial interactions in communities. This work was led by Aarti Ravikrishnan, who was my PhD student at the time, and Professor Meghna from computer science. And what did we do? We came up with a novel dynamic programming based enumeration to assemble reactions into pathways of a very specified size. So this was like a sort of input to the algorithm, producing a given target, another input to the algorithm from a given set of source molecules. So this is like the essentially the nutrient medium. And a given target could be actually growth or biomass or all the metabolites that are needed for survival or growth and so on. So, it's a very specific set of metabolites. The algorithm itself has two different phases. It starts with a guided breadth first search. I hope you recall breadth first search from your graph theory classes earlier. I did not cover them, but I am assuming that you have dealt with them in other courses, or you can always look it up. So, breadth first search essentially tries to systematically explore a graph by starting from a source node and then visiting every other node in a particular order. You basically assemble all the neighbors of a node into a queue if they have not already been visited or discovered, and you just repeat this process from the beginning till the end till you visit all the nodes in the graph. But here we have a small challenge because we use a bipartite graph representation. That is not really a challenge, but we want to account for the fact that you need whatever input metabolites are there for a given reaction node for that reaction to fire, so to speak, fire and produce the target metabolites, the product metabolites of that reaction. So, you have some A plus B plus C giving D plus E, let us say. You need A, B and C at the same time within the cell for the reaction 1 to happen and produce D and E. This sounds simple, but this is exactly the problem that we are trying to handle by a new representation, not using the substrate graphs, but using bipartite graphs and predicting pathways of a particular size and so on. We have implemented this in both Python 3.6 and 2.7. Some key features of the approach: this requires only the topology of the reaction network and does not require things like atom mapping, which is much harder to obtain. And so, which carbon is being converted to which what substituent on what carbon is being removed and things like that, right. So suppose glucose splits into two pyruvates, right. So, which carbon went where, right. There are two molecules of pyruvate that come out of one molecule of glucose at the end of the pathway. So, which atom went where. So, these are things that are typically covered in atom mapping, and this requires manual oversight, which means that it is not often scalable. You can pull these data from a database, but it can be challenging. We want it to be simple and scalable to large metabolic networks. That is really critical, and it needs to efficiently handle cyclic and branched pathways. Typically, cycles are a pain when it comes to graphs, and a lot of algorithms start going in circles. But jokes apart, a lot of algorithms are not readily able to adapt to cyclic graphs and so on. And we also need to be able to examine multiple alternate routes of conversion. So, this tells you a little bit more about an organism's ability, and we will not be using this extensively today, but this is another important feature of MedQuest. How do you handle community metabolic networks? You start with the directed bipartite graph of a single organism. So, this is the bipartite graph of a single organism, and this is the bipartite graph of the second organism. You now connect the graphs of individual organisms through a common pool of metabolites based on the overlapping sets of exchange reactions. So, it seems that F is being secreted by both organism 1 and organism 2. So, you say that F2, F1 or essentially F is being secreted by organism 1 and F can be taken up by organism 2. Both organisms could also secrete and uptake, right? These could be like bidirectional reactions and so on, for example. Right. So, this is a simple transport reaction that enables the exchange of B and F from organism 1 to organism 2. So, reaction 1 here, the exchange reaction 1 here is responsible for the transfer of B, the transport of B from organism 1 to 2, and this one is responsible for the transport of F from organism 1 to 2. So, what are the inputs to the algorithm? The input to the algorithm is a directed bipartite graph derived from a given metabolic network, a set of seed metabolites S, it is just a set of metabolites, and a set of target metabolites T, and an integer beta which bounds the size of any pathway that is generated. So, the essential question is, given G, how do you convert S to T within a particular number of steps? What are seed metabolites? This is typically a stumbling block. It's often difficult to accurately estimate this. This obviously includes whatever source metabolites that you are looking at, but it also includes cofactors and coenzymes which are typically always present in the cell. And typically, it is good to use at least a minimal medium, because you know that the organism survives in the conditions that you are talking about. So, what is the minimal medium that is required for the survival or growth of this organism in that under those conditions, right? So typically the medium in fact determines the conditions. So, it tells you what carbon sources are present or absent and so on. So, under these conditions, what is the minimal medium that is required? And just a quick walkthrough. So, we start with a directed bipartite graph derived from the metabolic networks of seed metabolites, and you can just recall the bipartite nature here. You have two types of nodes, you have circles and squares. So, circles are basically metabolites and squares are reactions, and what you see is that you need both input metabolites for producing an output metabolite. So, that is the main aspect that we are trying to capture here, so that we have more reliable predictions. So, the output is what is known as the scope of metabolites, which is essentially all the metabolites that can be produced within the cell under the current conditions. And these conditions typically for us, because we are not integrating any expression data and things like that, it just typically means what is the medium available. So, under this nutrient condition or under these nutrient conditions, this is what is possible for the cell to produce. And it also gives us output the reaction set that can be visited in a pretty BFS style, breadth first search style. So, in this case you look at the simple example, if M1 and A1 are present, R1 fires, so to speak, and M2 is produced and visited. But if M1 is present and A1 is not present, then R1 cannot fire and M2 is what we call as stuck. So, you have a bunch of metabolites that are producible and a bunch of metabolites that are stuck in a metabolic network based on what other metabolites are present. Right? If I do not have glucose, I cannot make pyruvate. Well, then the cell can't even grow, but essentially you are looking at examples such as these. If I have A, I can produce X. If I do not have A, I cannot produce X. And now we need to assemble these reactions into pathways. So, let us look at we build a table, and here is a simple metabolic network again. So, here you have all the metabolites on one side. So, these are the rows of this table, and the columns are all numbers up to beta, right. So, how do you build pathways of particular sizes? So, with 0 reactions you can produce any input metabolite. So, this is basically the seed set of metabolites, and there is no way to produce any of the other metabolites in 0 steps, and that is what is indicated by this null set symbol. And what you see here is that there is no way to produce M1 and A1 with a single reaction 2, but M2 can be produced by a single reaction R1 in one step. Okay. And what are the sets of 2 reactions that can produce any of the metabolites? So, M3 can be produced by R1 and R2 In fact, there is only one way to produce M3, so you can just produce it using R1 and R2, but it is the same for producing M4 as well. That is what you see here. Now let us look at M8 and how many reactions it requires for its production, right. So you start with M1 and A1, they are the seed metabolites, and you go to R1, and then R1 produces M2, and M2 is able to produce R2 and R3, but we do not need to worry about this, this part right now. And then what you see is R3 produces M5, which can produce M8. So with R1, R3 and R6 you can produce M8, and that is what is reflected here. But if you see, there is no M7 in this table, and then because there is no way to produce M7 using 0, 1, 2 or 3 reactions, right, you need more than that, because with just R1 and R3 you can produce M5, but you cannot produce M7 at that point because M4 will not be there. If R2 also happens, then of course you can produce M4, and then reaction 5 works, and then you can produce M7 as well. But you need at least R1, R2, R3, R5 for producing M7. You require at least 4 reactions for producing M7. So hope you are getting the hang of this kind of this approach. On microbial communities, again you can run the same algorithm, it just becomes a little larger and a little more challenging. So we looked at a consortium of Clostridium, Cellulolyticum, D-sulfo, Vibrio vulgaris and Geobacter sulfur-reducens, and we constructed a pretty large directed graph out of this. The network contained more than 14,000 nodes and nearly 29,000 edges, and the size of the scope was huge. 1,000 odd metabolites could be produced in this community, and we looked at pathways of size 20 to all the metabolites within the scope of cellobios and other metabolites, because this was studied in one of the papers previously, and we found that across the paths that we predicted, acetate, pyruvate and ethanol were very frequently exchanged, as was previously demonstrated in another study. Essentially, the MetQuest approach can be readily scaled to large communities, and they can be modeled very nicely as bipartite graphs, and you make predictions on what metabolites can be produced, cannot be produced and so on.